Do you need smart and hassle-free laboratory workflows? In this video, we will show you how to achieve this using sequence by barcode. Normally, when you create a sequence list, you require certain information. This is, for example, information about the sample name, the method you want to use on the sample, and the location of the vial. All this information is gathered as normally manually added to a so-called sequence list. Most of the information are most times already present in some kind of database. For example, a lab information management system, a so-called LIMS system. This information can be linked via a barcode with the sample. On the Gerstl MPS robotic system, the barcode is read using the Gerstl SID2 barcode reader. After the information is read by the SID2 barcode reader, the information is processed by using the so-called sequence by barcode. Sequence by barcode can be used in two different modes, day shift or night shift mode. The day shift mode is the so-called single mode. In this mode, the MPS robotic takes one vial, transports it to the SID2 barcode reader, reads the information, creates a one-lined sequence list, processes this list, and the information is stored. After that, the next vial is read, and this continues up until the last vial. The day shift runs in a single mode. One sample is scanned at a time and is processed in a single run. This allows for changing the order or change even the priority of the samples. In addition, you can add so-called QC standards or even add automated calibration lists. This allows highest flexibility in the lab. In the night shift, we are working in the so-called batch mode. In the batch mode, the MPS robotic takes all sample vials from a tray and reads the information stored in the barcode. This information is then added to a sequence list. After that, the sequence list is processed. In the batch modes, we are scanning all samples and working in so-called batch sequences. This allows for the best possible overlap using the prep-ahead mode of the Gerstl Maestro software. In addition, you can also add QC standards or use automated calibration series. In this mode, we can reach the highest possible throughput of the system. In this picture, you can see the inside of the SID2 barcode reader with a vial with the barcode attached from the position of the optic of the barcode reader. As you can see, by the aligning of the mirrors, there are almost no positions where a barcode cannot be read. If there is really the case that a barcode cannot be read, this information is stored, logged, and the vial is skipped. In the last minutes, we were always talking about vials, but of course it is also possible to use TD tubes, which are delivered with barcodes attached by Gerstl. With sequence by barcode, it is possible to process 1D and 2D barcodes to create automatically sequences. With the database approach, using the information that is stored also in the LIMS, it is possible to reach the highest flexibility or throughput depending on your needs. It is also possible to automatically add control standards or calibration series. So, make your lab smart using Gerstl sequence by barcode. Watch also the other videos from this series and remain curious.